Hey all, this is Matt. Since I was unavailable to present my paper of the week this week, rather than just put a few summaries into a slideshow and send that around, I thought I'd try doing a virtual presentation of this, which is easier said than done. It's a lot easier to speak live than to try and record yourself. I think I'm on like take 3 or 4 at this point. But anyway, I'm just going to power through this and hopefully share just some of the highlights that I see from this paper and be able to put this out there for further discussion as you're interested. So I picked out the follow-up paper actually from my last paper of the week, which was back in April, which is the next installment of the exact exome sequence analysis, and this is focusing on rare copy number variation. So let me go over to my slides, which I will post in the on the team site if anyone wants to access those and review any of this in the future. So again, the senior author on this is Sean Purcell, but of course includes a lot of the other senior authors from the other work on the exact. So there's a lot of commonality among them. This slide is essentially the paper in brief. So I tried to call out what I thought some of the key methods were, some of the primary observations, and then a take-home message. And I'll go to the next couple of slides to provide a little bit more detail on some specific results in the paper. So in terms of the methods, we had looked into calling copy number variation from exome sequence data back in the 2012-13 timeframe when we were collaborating with Gonzalo Abecasas and Dan Hovelson at University of Michigan when we had the NEVA project. Remember the exome sequencing that we had done on the East Asians, and we were investigating those copy number variations for the ADME genes, which this could be quite important for. And the methods that Gonzalo was using at the time didn't seem to do a very good job. We really didn't trust the kinds of gene copy number results that we were seeing from that. So we make some reference to it in the paper, but we essentially abandoned that and certainly abandoned any effort to use the results from that. In this paper, they're using a method called XHMM that was essentially published right around the same time as we were looking into this. This was published in the American Society of Human Genetics journal, American Journal of Human Genetics, back in 2012, and so they seem to feel that this was a reasonably robust copy number calling algorithm from exome sequence data, and this is what they applied. So it's a combination of getting the average depth of coverage from any given target with GATK, and then parsing that through this XHMMM methodology to call copy number variation at the target level. So in this instance, when we say target, we're talking about the capture probes for any portion of DNA that's being sequenced. So just to highlight, so it's roughly 60,000. They started with the same subset of 60,000 plus that were part of the previous exact paper, but just after some additional QC, the sample set decreased a little bit. But you can see that it's more or less apples to apples when comparing these subjects. So all of the sample level QC that had gone into the previous exact paper was the starting point for the subjects in this paper. They do report a little fun fact that it took about 1 month of compute time with 800 gigs of RAM in order to go through this process. So it's no small feat to be able to compute this. They didn't indicate how many CPU hours or years were involved in that month of compute time. They had identified about 126,000 copy number variants total. They threw out the common copy number variants and really focused just on the rare variants for the kind of intolerance estimates that they wanted to include in this paper. They removed any genes where at least half of the exons weren't well captured. That is, it met the criteria for being able to call copy number variants. In the final analysis, they had just under 16,000 genes from the gen code, I think version 19, that were in which they had at least one copy number. And when they talk about copy number variants in genes, it could be one or more exons that are affected by that. So that gets a little bit ambiguous when you look at their summaries. And 54% of array-based copy number variants were also called by the exome sequence. So they did a big QC here and tried to assess the repeatability between calling copy number variants with genotyping arrays versus from the exome sequence. And what they found is, I guess first of all, with the array-based, it's not particularly powerful for calling small copy number variants. So there aren't many instances where you could call, for example, just a deletion or duplication of a single exon with the array-based methodology. So so those large copy numbers tended to be more robust for the array-based, and they cited some statistics about higher coverage between the two methods. So basically what they saw is that 54% of the time when they called a copy number variant with the array-based genotypes, they also called the same copy number variant with the exome sequence data. Those two overlapped. So that's only half. That's not great. Conversely, 25% of the time when they called copy number variants and one or more exons from the exome data, the sequence data, did they call it from the array? So as suggested, you're not going to call as many of the relatively small copy numbers that you could potentially pick up with the exome sequence data. In terms of the carriage statistics, about 70% of people carried at least one rare copy number variant. On average, a person carries 0.81 deletions and 1.75 duplications. So essentially, you're twice as likely to have a duplication at a gene than a deletion at a gene. Only 16% of the rare copy number variants were more than 100 kb in size, as called from the exome sequence data. Of course, we're focusing really just on the target sequence for these capture arrays. And interestingly enough, and consistent with some previous array-based results, they saw that females were more likely to have differences in copy number, either deletions or duplications, than males. That was highly significant. And the gene, so getting down at how much data underlies the ability to draw any inferences from either the negative selection for copy number variants. On average, any particular gene only had about 3.1 individuals with deletions and 6.6 individuals with duplications. So that's not many average CNV events to observe to try and draw inferences about whether you're seeing a dearth of copy number variants that would indicate intolerance. So, and I'll come back to this in a minute. And finally, so the copy number intolerance scores calculated in a manner similar to RVIS. That is that they fit a regression model, here a logistic regression model, where they related the size of the gene to the frequency of the copy number. They also included in the model other things that might affect that aside from gene length, which would be read depth, GC content, sequence complexity, and segmental duplications in the region. Just coming back to an earlier idea, the copy number intolerance estimates are probably pretty noisy because you don't have that many events per gene, even in a data set of 60,000 individuals. So I think that it's going to take much larger data sets or perhaps better copy number calling methods to be able to get the same level of precision that they're getting even for the loss of function estimates, which themselves are also still a bit noisy. Missense variants are probably far more robust because there are more observations of those in these data sets. In terms of some of the key observations in terms of the functional consequence of these genes being predicted to be intolerant to copy number. So in most tissues, it's the highly expressed genes that are less tolerant to copy number variants. That is that they tend to be constrained. with, as you can see, substantial heterogeneity among tissues. So in this figure, this is from figure 3, they have estimated the enrichment of intolerant genes that are highly expressed in each of these tissues. And for 17 of these tissues, you're estimating that there is that the highly expressed genes are more likely to be intolerant to copy number variants. You only have a few different tissues where you're estimating that there's going to be an excess or an enrichment of copy number tolerant genes being highly expressed in those genes. Similarly, they took those per gene estimates and they ran it through just a gene ontology gene set enrichment analysis to look for particular classes of genes that appear to be intolerant. And so these are just in figure 3b some of the categories that they'd identified. So a lot of brain-related pathways or classifications seem to be involved. And you can see a few other methods or a few other categories as well. Similarly to what I had done in the April presentation, I also went ahead and looked at for the copy number intolerance percentiles. Essentially, do we see much differences in these intolerance percentiles as you look across what can be some pretty interesting categories of genes? So here we've got all genes, and as you'd expect, the median is at the 50th percentile, and it goes from 0 to 1. And then you have the genes for which there's at least one GWAS association, at least one Mendelian disorder reported for that gene, whether or not the gene is druggable, whether or not that gene is one of the core 300 epigenetic reader-writer eraser genes, whether that gene is the target in a genomic regulatory block, these ultra-conserved regions that are really important in development, whether or not the gene is in clinical development, and whether or not the gene is the target for a drug that's marketed. So upper left is the percentile for the missense z-score from the original exact paper. On the upper right is the loss of function z-score percentile. And as we talked about before, some of these categories of genes are really highly constrained, really intolerant to loss of function variants. In particular, this class of epigenetic genes are really intolerant, and you see a nice pattern with the GWAS and the genes underlying GWAS and rare diseases. And we even see the enrichment amongst those under clinical development and marketed. Down below, we've got the 3 plots for the 3 different types of intolerance scores that are provided: an overall intolerance score, an intolerance to deletion score, and an intolerance to duplication score. And really, we don't see really strong patterns for any of these. We don't really see any enrichment of intolerant genes amongst genes that have GWAS or rare disease associations. Really nothing differentiating the druggable genes. Maybe a little bit of a trend towards more intolerant genes amongst the epigenetic genes. Really, GRB provides us the strongest signal, and again, just a slight trend for those under clinical development or marketed drugs. When you break it out by deletion versus duplication, we actually see somewhat stronger patterns for I would say the intolerance to deletion overall, but again, it's not so strong. My interpretation of this is it's probably a couple of different things. One is, I think, just the noise, the lack of precision in estimating these intolerance for copy number, because there are so few copy number events observed. And there also may be something really biological underlying this, suggesting that it just may not be as deleterious to protein function when you have modest numbers of intolerance of copy numbers. I looked at the correlation between these 5 different measures of intolerance, so missense, loss of function, and then the 3 copy number. The correlation coefficient, if you look at the loss of function intolerance versus the copy number intolerance measures, are pretty modest. You get the highest correlation with the deletion and loss of function. That makes sense intuitively. Although with a correlation coefficient of 0.25, the R squared is only about, it's less than 7%. So we really aren't seeing a very strong relationship between them. And again, I think it's a mix of, perhaps there's some biology that explains that, population dynamics, the impact that these have, as well as just the noisiness in these estimates. So just wanted to highlight that I have updated the spreadsheet that I had created before and had placed in the wiki. Let me pull that up here. So here is the, let me go back to the wiki page. Whoops. All right, so here's the gene constraints wiki page. Refresh that, because I've done some editing here. There we go. So I've updated this gene constraints wiki page. It now has a link to the latest spreadsheet, which has all of these intolerance scores, as well as some other things that I've added over the past several months. So if you want to be able to access that, you can find it here. And this page here provides a description of each of the fields that are in that spreadsheet. So you can go through that. I've also updated some of the text in here, so it has links to all the papers that are included, and some of the key findings that I've summarized here in this presentation. So that was way longer than the 5 minutes that would typically be a summary of a paper in a paper of the week. But for the 1 or 2 people that actually listened to this and followed it to the end, thanks for listening. Feedback welcome. Be interested to hear how effective or ineffective you think this method of sharing a paper of the week when we don't have time to review it as a group is or isn't. But anyway, thanks.